Hey, what's up, guys? This is Audie from ShastryNation, and today we're going to be learning about cloning. Alright, so now that you're back from the intro, I guess we can go ahead and get started. Alright, so cloning. Oh, I should get... one second. Yeah. There it is. Oh. Cloning. Okay. So cloning is, it's what you think it would be. Like, cloning is just basically you take, whatever you need to, and then you make a copy of it. So it's like the traditional, I guess, sci-fi esque thing of cloning, basically. But now we can actually do that. We can actually clone stuff now. Now I don't know if you guys read the news, but I think a couple of years ago, it was a couple of years ago, I think 2010 or so, they actually cloned a sheep, Dolly the sheep. They cloned it, and we'll talk about the types of cloning. There's multiple types of cloning, and they're used for different reasons and things like that. And there's also a difficulty level and things like that with each of those. So this video won't be too long. I know I missed the last one because I just had a lot of stuff on my hands, going on. As you guys know, I am building a website. Basically, I have I think 25 articles on there right about now. So if you guys, should go and check those out because those are pretty cool. I love writing stuff on the website. It's pretty awesome. So that's why I haven't been really making that many videos and things like that. I am going to try to come out with an esports video, probably I think tomorrow or the next day or Wednesday or so. I know I haven't been, making a lot of gaming article news things a lot, but there hasn't been that much going on. So before we waste a lot of time by talking about all this type of stuff, why don't we go ahead and get started? Alright, so I guess we can start out with why do we need to clone? Alright, so basically cloning first of all, it's cool. I don't know if you guys know that. It's pretty freaking awesome. Cloning is actually really cool. So basically we need it for, to do things, to cure things. We're going to actually talk about this one thing where they can actually grow organs and things like that. And you can actually cure stuff with it and other things like that. So, there's a lot of stuff that you can do with cloning, and there's a lot of uses for it, but it's also really difficult to do so. You know what I mean? We have the technology, but we need to develop it more to actually make it useful for us. Yeah. All right, so let's go ahead and talk about the types of cloning. And we will get this episode of biology on the way, and we will get done with this pretty quickly, actually. Alright, so first one we're going to talk about is recombinant. If you guys hear the TV, it's my mom and my sister. They're watching TV in the background. So that's what that is. I'll try to edit it out, but if it's not going to edit out, then you're going to listen to it. Alright, it's DNA cloning. Okay, so recombinant DNA cloning. So basically what they do is they put bacteria, they put DNA from a eukaryotic cell into the bacteria, and they make it, they make it form something. Basically, so we'll just write it down. So basically they put DNA into bacteria to get it to make something. Make the protein. So basically they're gonna put DNA into a bacteria and to make it into a protein, basically. And they use this for example, for insulin, insulin shots and stuff like that. Basically what happens is, and we'll talk about this when we go to cell communication, because diabetes is really a cell communication problem. It's a cell communication gone wrong type thing. And so basically, what happens in diabetes is in type 1 diabetes, the people aren't the patient or the person isn't actually making insulin. And so, we try to counteract this by actually making insulin, artificial insulin. And how scientists and doctors actually do that is by putting DNA into bacteria and then getting it to make insulin, so that it can, go in and get the cells to actually take in the glucose, so they'll have lower glucose in their systems, and that'll stop a bunch of the problems that they have as diabetics. So there's a number of problems with this. The fact that, if you haven't watched the DNA expression, the gene expression video, and all the types of restrictions and stuff like that are put on DNA, I would go and watch that, because a lot of the stuff we're going to use is going to be used in this video. I think that was a couple videos back, so it shouldn't be too far down. I would go and watch that one right now. You can pause the video, I guess, right here. Okay, so now if you've already watched the video, we will go ahead and continue. So basically what happens is that, when basically the problem is that when you put in DNA, when DNA is, if you can remember back to that video, when DNA is being, cut out and things like that, like a certain gene is being cut out of the DNA, basically it's being copied and things like that, we have a number of parts of the DNA that are being restricted, you know what I mean? So we'll have like exons, introns being restricted, and it'll cause the DNA to, it'll cause the gene to perform exactly what the cell needs. Now, the problem is that bacteria don't have exons and introns, so the introns won't actually be cut out. And so that's the problem. We have to figure out a way to get in, the DNA, but also like cut out all the parts that we don't need. Because if we don't cut out the parts that we don't need, that'll make a completely different protein, and maybe it'll cause some, reaction in the patient. We don't want that. So basically the solution to that is that they actually try to just take the mRNA from the cell. So basically, you'll have all the introns and everything is going to be cut out and everything's going to be regulated. So they're going to take the mRNA somehow by doing a bunch of chemical reactions and stuff and separating the mRNA from everything else in the cell. They're going to take that and they're going to put it into the bacteria. Another problem with the bacteria is it's not going to take up eukaryotic. We're going to have a difficulty for it to take up eukaryotic genes, because it doesn't. Eukaryotic and prokaryotic genes and DNA don't. It doesn't mix. We have the same type of DNA. We can get it to work, but it's going to be a little difficult to actually do so. That's another issue with this. But, for the most part it works, and that's why we have insulin shots and things like that. Yes, we could use like the polymerase chain reaction. If you haven't watched my DNA tech video, I would go and do that. So we could use something like that to make all the proteins and things like that, whatever we need. But, this is also another way that you can make insulin and other things depending on what the patient or person needs. Alright, so then we have, I guess we can move on. So let's use a blue. Alright. So then the next one we have is reproductive cloning. So reproductive cloning. Okay, reproductive cloning. So basically this is what, you would actually think would happen when you clone something. Basically what happens is that you basically put DNA from like a host, from whatever animal you want to replicate or like clone. You put that DNA into a zygote. No, not a zygote, but like a diploid cell, like basically an egg. And basically what'll happen is that the egg will take up the DNA of the other, of the DNA you put into it, and it'll make like a carbon copy, I guess, of the organism that you wanted it to make a copy of. The DNA is going to be expressed in the same way and things like that, and so it'll produce a very, very similar, organism. And this is actually what they did for Dolly, the sheep. Basically, they just took, they put it into an embryo, and they put Dolly's DNA into an embryo, and they made another Dolly. Basically, they made two Dollys that are exactly the same, genetically and phenotypically the same. All right. So basically, they put DNA into an egg. That's about it. DNA. Put DNA into an embryo. That's what I'll say. Embryo is what I'll say. Okay, so the example would be Dolly the sheep. Sheep is plural, but I don't know what the singular is actually. So, all right. Therapeutic cloning. All right. Now this is the really cool one in my opinion. Okay, and I could see the implications. So therapeutic. And with this, we're almost done with our video. So therapeutic cloning. Okay, so therapeutic cloning is going to be, the cool thing where you basically like grow organs and things like that. Now this is obviously it's going to be the most difficult to do so. But basically the idea is that you can take up any cell from any part of your body, because every cell will have the exact genome, your exact genome, like your entire genome will be in every single cell inside of your body, right? So basically the idea is, like I already started to tell you, the idea is to take any cell from your body and rearrange the DNA. So let's say you needed a new, I don't know, let's say you needed a liver or something like that. Okay, so let's say you needed a liver. They could take a cell from your hand or whatever part of your body. They'll take a little bit of your cells, and with a lot of magic, notice how I said magic because it's very, very complicated. Sorry, it's very, very complicated how they do this. But they'll rearrange the DNA. So if I took cells from my hand, they would rearrange the DNA to show the cell the parts of the DNA that that make up my liver. So basically they're gonna make my hand cells, I guess, into liver cells, and basically they're gonna do that, and they're gonna grow another liver basically for me, and they can transplant it in. The problems with this, obviously you probably know the problems with this. There's a lot of stuff that can go wrong. First of all, that's an issue of actually rearranging the DNA. That's an issue. Second issue is the fact that it's really difficult to actually grow cells outside of a body. It's just weird. It doesn't happen. I mean, the only known example of cells going outside of the body is HeLa cells or Henrietta Lacks cells, and those are the only cells, human cells, that are being grown outside of the body. So basically, it's very difficult to do this. Now, another thing that they are planning to do, and is definitely possible. Let me go ahead and write down what this is. So they grow organs from other cells. Basically, what they're also trying to do, I don't know if you guys have heard of stem cell research, but basically stem cells are going to be like the first, when you're first formed as an embryo, you're going to have stem cells, which are basically like the original copies of the DNA. The first original copies of the DNA, and then everything else is going to be a copy of the DNA. And so basically, you'll start out with these cells, and so basically, the idea is that if you can take your stem cells from your bone marrow, because your bone marrow will have stem cells to make, B cells, and then your thymus will obviously have the T cells for well, we'll get into B cells and T cells when we go into immune system. But anyways, your bone marrow will actually have the stem cells that you need to make your immune system cells or red blood cells or anything basically that you need to make. Because you need to use your stem cells because there's always a chance for mistakes to happen if there's too many generations. You know what I mean? Like it's more common for a mistake to happen 10 generations down the line, a fatal mistake to happen 10 generations down the line with cell generations. So it's obviously always going to be replicated and things like that. Versus having the DNA being the cell being made directly from the source that it's supposed to be made from. So basically that's why they have they try to use their stem cells. So stem cells are basically, like I mentioned before, they're going to be the main, the first copies of DNA. So they're going to be the true copies of your DNA. Everything else is going to be filled with mutations and stuff like that, because obviously mutations will happen. If you want to know more about mutations, I would go and watch my mutations video. It's I think 20-30 minutes long, so it's a lot of stuff on mutations that can occur. But basically they're trying to use stem cells to make organs and bones and whatever else they need to, and basically they're going to try to transplant it into you. Now, obviously, you guys know the problems with this, things like that. And they're actually, it's pretty interesting. They actually already have a 3D printing, 3D bioprinter machine type thing. I don't know if you guys know what that means because I just explained it really incorrectly. But anyways, basically, it's just like a 3D printer, but you can grow organs and whatnot into it, and it's really cool. I think that's really the future of medicine. That's just my opinion. Being able to, not having to wait for a transplant. Okay, for if someone had heart disease or something like that, they wouldn't have to wait for a transplant. They would just be able to grow their own organ. And then it would work for them because it's their own DNA that's being used for their organ. There's always a chance of rejection when you get transplants and things like that, especially, involving major organs like the heart or the liver or the thymus or, anything. Basically, any organ is there's always a chance of rejection by the body, and that could be fatal. Because your immune system will basically start to attack the organ, and you will start to die. So there's a lot of gray area with therapeutic cloning, but it's definitely, I think it's developing. So we'll have to see what happens with therapeutic cloning. I think it's really cool. I think it's a really sound idea, but I don't know if it's really probable right now. I mean, they are making stuff that happens, and I am going on forever about this, but you can tell that I really am passionate about therapeutic cloning, I guess. Alright, so I guess, that's about it. There's not much more that we can actually talk about with the types of cloning. I mean, I tried to make this video as short as possible, because I wanted to make it short, but basically this is about it. So if you really wanted to, basically know the 3 types of cloning, just know them, I think there's another one, but I'm not totally sure which one that is. I don't think we learned it. I don't know. But anyways, these are the 3 main types of cloning. Obviously I think therapeutic cloning is probably the coolest. But, the other 3 are also pretty awesome, too. And, hey, it's cloning, so cloning is really cool. But, yeah, that's about it, Just know these, know them pretty well, and understand them. And, maybe if you want to, go ahead and read about some of them. Because there's a lot of articles and journals and scientific papers and things like that on all of these. All right, so I guess I will leave you guys at that, and I will see you guys on the next one.